Kia ora koutou. Thank you, Nick. Well, these species are probably familiar to everyone in the room. They represent some of the worst and some of the most ubiquitous invasive weeds in the country. They were all introduced here intentionally from faraway places. And they present a big conservation threat, but they also form the backbone of the country's agricultural and forestry sectors. So ironically, we want these species to succeed, but we don't want them to be too successful. So, there we go. And sometimes, as we know, they do become too successful, and they escape their intended boundaries. And we think this might be because they've come from this other place where they were kept in check by natural enemies. And they've come to this new place where potentially those enemies don't occur or they have less impact on these species. And this is the whole idea behind biocontrol, right? So when these species become very successful, we go back to the species' home country, we get these enemies, we bring them back to control them. It sounds pretty crazy, doesn't it? But biocontrol and the success of biocontrol really does, it's evidence for the importance of biotic interactions in shaping invasive species success and impacts. And so that's what I'm going to talk about today, is how invasive species success is mediated by interactions with other organisms. And first, I just want to say big thanks to so many people and acknowledge all the wonderful folks who have helped out here. On the top, we've got our PIs, postdocs, and students on the right there, and loads of other people. And I could spend about 15 more minutes just showing you faces of people who've helped, and actually large equipment and machinery that helps with all the research that I'm going to show you today. But in the interest of time, I'll just say that the work I'm going to show you is the result of many, many helping hands and minds. So the first project I'll talk about is Project 6, Achieving Bioprotection in New Zealand Ecosystems. So we set up a large multifactorial experiment to explore how interactions between specifically invertebrate herbivores and soil biota affect invasive species success. That's my partner there, Warwick Allen, who did all of this work with me. So I'll just tell you a little bit about the design of this experiment. There's kind of a lot to take in. So what we did is we created and designed these 20 unique little mini ecosystems, little mesocosms. These communities, they varied in their proportion of exotic species, ranging from 0 to 100%, with some mixtures in between, and their proportion of woody species, from 0 to 60%. Each community was made up of 8 species, and they were chosen from a pool of 39 native and exotic species, all that co-occur, particularly in the Canterbury region. So I told you that we did these manipulations of the soil biota and the herbivore communities. To manipulate the soil biota, we went out into the field and we collected truckloads of soil from places where all of these species occurred. We mixed up all that soil, we brought it back to the greenhouse, and we grew plants in those soils for about a year to culture the specific microbial communities that would typically associate with all of those species. After about a year, we harvested them, we retained all that soil, and we made up these soil mixtures to put out into our pots in the field. So we had what we called home soils and away soils. A home soil would consist of all of the soils from the species that were grown in a specific community. So presumably would contain the generalists and specialists, enemies of those plants. An away soil would consist of 8 soils from 8 species that did not occur in those communities, and presumably only occurred the generalist enemies. Warwick, the insect whisperer, was in charge of the herbivore additions, and he went out into the field and collected loads of insects. He also, I think, got some from some people in this room. He reared them in the lab, and he introduced them to all of these mesocosms. So you're probably wondering why they all look like ghosts. Those are mesh cages that are around each of those pots, those plant communities, and those are meant to keep those herbivores either in or out. In all, he was able to get 20 herbivores established. These were a mix of exotic and native species. There were some agricultural pests. They were mostly generalists, and they had a range of feeding styles. There were root, stem, leaf chewers, suckers, and miners. And Warwick did a number of surveys through the extent of the experiment, and he extrapolated biomass estimates and things like that. So we let these communities grow for about a year, and then we harvested them. And one thing that was very apparent is that the exotic plants had very, very big impacts. This is just one of the communities that we have where you see these two giant exotic plants, and there's a tussock grass that's literally hanging on for dear life. I think it's trying to escape from the pot there. And so we found this across the board. Exotic plants were very big compared to the native plants. This is just a graph showing you the above biomass in those pots. They were quite large. They were also quite competitive. Sorry about the graph is looking a little bit funny. I'm a Mac user. Enough said. And so as you can see, this is just a graph showing you how the richness in these pots declined in these exotic pots. And please take a second to get used to this, the format of this graph, because a lot of the graphs will look like this. On the x-axis, we have the proportion of exotic plants ranging from 0, so our native communities on the left, all the way to 100% exotic to the right there. On the y-axis, we have our response variable here. It's just the number of species that were in the pots at the end of the experiment. And as you can see, in these native-dominated communities, we started with 8 species, and we often finished with 8 species. But as you add invaders, you see that richness really declines quite sharply. So they were very competitive, even among themselves. So there were some differences among invaders in their competitive ability as well. So exotic plants had big impacts in communities. They were very large. They were strong competitors. So far, not a lot of surprises, right? But the way that we designed this experiment really allowed us to tease apart the effects of biotic interactions on these impacts. And so that's what I'm going to tell you about. But first, I'll just talk about some of our hypotheses. So there are a number of scenarios that could happen here. We might expect that exotics make weaker interactions with insect herbivores and soil biota, in which case then the natives would be harmed to a greater extent than the exotics, and then we'd hypothesize that exotic plants can outcompete the natives in those communities. Alternatively, we may see indirect effects where the exotics make stronger interactions with enemies. Those enemies accumulate, and then they spill over onto the natives, giving the exotics another advantage, and they may, in this case, also be able to outcompete the natives. So we were able to test these hypotheses with this design, and I'll tell you about that starting with the herbivores first. So exotic plants did make stronger interactions with invertebrate herbivores. Again, this is that same format of the graph here. We see that herbivore species richness increased along our exotic gradient. There were more herbivores, and there was a greater biomass of herbivores in these communities. But did this translate into increased competitive ability? Well, not really. So on this graph here on the left is showing you, again, the same graph I showed you earlier, native and exotic plant biomass. We still see that exotic plants are larger in these pots. Even though they're accumulating all of these herbivores and these herbivores are doing damage, they're still much larger. But what this graph shows you is this difference with the herbivore treatment. So you can see that herbivores had no effect on native biomass, but they did have a strong effect on the exotic plant biomass. So there's a 30% reduction in their biomass in those herbivore plots, but they still were incredible competitors, as you can see here in this graph on the right. So what you're looking at is you can see these dotted lines, and that's just the proportion of biomass that we planted these plants into in the community when we started. So as you can see, there's 25%, 50%, and 75%. And the dots that you see above that, that's our finishing biomass in these pots. So the exotic plants did really, really well. They took up a much greater proportion of biomass in these pots, despite the fact that they made these really strong interactions and were quite damaged by herbivores. So exotics did make stronger interactions with herbivores compared with natives, but these enemies did not spill over onto the natives, and these stronger negative interactions did not reduce their competitive ability in these plots. But so how did they do it? How were these exotic plants still so competitive? Well, another thing that we found is that there was higher herbivore biomass in communities that had exotic plants with high specific leaf area. This is a trait that's associated with faster growth, higher tolerance to herbivores. So it suggests that maybe there's a trait explanation for this, for what we saw, that maybe these plants are just able to grow a lot faster despite this herbivore pressure. So next, I'll tell you about the soil biota and what we found there. So remember, we grew these plants in home and away soil. And what we saw is we saw a big effect of our home soil treatment on these plants. So when plants were grown with specialist and generalist enemies, presumably, their biomass decreased quite a bit, as opposed to these away soils where they did quite a bit better. But there was no effect of plant provenance. So natives and exotics were both harmed to a similar extent by our soil treatment, that home soil treatment. But you know, you can really only say so much about these kinds of results. These are plants that are grown in bulk soil. The effects that we're seeing, they could be due to pathogens in those soils. They could be due to poor mutualists or any number of things. So another thing that we did to try to tease apart the effects of soil biota is we did barcoding. We sequenced all of the fungal communities in these plant roots, all 1,152 of the roots. We then used taxonomic functional databases to assign function to our sequences. And then we also measured PLFAs and NLFAs to measure the fungal and bacterial biomass in the soils. And so, another funky graph, sorry. So what we found was we saw that pathogen abundance increased along our exotic gradient. So again, here's this. We're looking at these communities. As you increase exotic dominance, we see this increase in relative pathogen abundance in those communities. And we also saw a sharp decrease in mutualistic fungi in these exotic dominated plots. So they're making stronger interactions with enemies, once again, in the soil. But again, and also, if we look at the plant, at the individual plant level, we also saw that exotic plants supported more pathogens, more sequences in their roots that were pathogenic to these plants. But did this translate to reduced competitive ability by the exotics? No, the exotics, this is the same graph I showed you before. Exotic plants were really big. Again, in spite of the fact that they're accumulating all of these pathogens, they're still quite competitive and quite large. So to try to understand how the soil biota might be driving some of these effects, what we did is we looked at the proportion of pathogens that were shared in each of these communities between natives and exotics. So if you have a given community, we looked at how many pathogens were associating just with natives, just with exotics, or shared between the two. And what we found is that there was quite a bit of sharing of pathogens. Now, this is a really ugly graph, so I'm just going to walk you through it slowly. So what we're looking at here is how the proportion of shared pathogens changed along this exotic plant gradient. So you see it's a hump shape, and it's a hump shape because we're at zero and 100% exotics, right? We can't be sharing any pathogens. But as you increase the number of exotic plants in the community, you see an increase in the proportion of shared pathogens. But what's interesting about this graph is if you focus on that gray band there, what that is it's a simulated null model. It's telling us the sharing that would occur by chance if natives and exotics were completely equivalent in their ability to share pathogens. So we ran that model 1,000 times, and we came up with this gray bar. And so what's interesting is that when you look at the real data over this, you can see that our black line in the native-dominated plots is below that null model expectation, where in the exotic-dominated plots, it peaks and it comes up over. So what it's telling us is that these exotic plants are more likely to share pathogens than you would expect by chance, where the natives are less likely. So these natives appear to be more specialized in their interactions, and these exotic plants tend to be more generalized. So finally, the last graph I'll show you from this project here is we're trying to figure out whether or not there's any correlation between pathogen sharing and plant success. So we use this equation here, the relative interaction intensity index, which is just gives us an indication of competitive ability. So any of the dots that you're going to see in that blue area above the zero line indicates stronger competitive ability by natives. Anything below that zero line in the peach indicates a stronger competitive ability by exotic plants. And so if we look at that and we look at this compared to the proportion of shared pathogens, what we see is if you look at about 30% of shared pathogens there, that's where that line is. That's where natives are always doing worse. So we have some variation in competitive ability in these different plots, but it really declines once you are sharing about 30% of the pathogens, and in that case, exotics win every time. So this just suggests that there's some correlation between pathogen sharing and this plant competitive ability. So just to summarize the results from this project, exotic plants had strong impacts on native plants, unsurprising. Exotic plants supported more herbivores, more fungal pathogens, fewer mutualistic fungi. Exotic plants were more successful in spite of this, in spite of the high herbivore and pathogen loads. And exotic plants shared pathogens with natives, which did correlate with exotic plant success in these communities. And so now I'm going to tell you a little bit about Project 7, some of the work that's been done in that space, looking at whether fragmented kauri ecosystems facilitate pathogen spread. And we know that kauri is in real danger. Its habitat has been shrinking and shrinking to under 1% of what it was once. Particularly now, it's under threat by the pathogen Phytophthora agathodicida. So I showed you in the last project that we saw these big changes in fungal communities, right, when we increase the proportion of exotic plants that are in a community. Well, Alexa Byers, who is a recent graduate of BPRC, she looked at fungal communities in kauri and in pine plantation soils, and she found a similar result. She found that fungal communities were quite different in these two different land use types, indicating that there may be some functional consequences of these changes in these forests as well. And then Kai Lewis, another graduate of BPRC, he did a soil assay, and he looked at the potential for reproduction of this pathogen in soils from kauri soils and from pasture and from pine soils. And what he found is greater reproduction in these exotic soils, again, suggesting that there is some reservoir potential in these exotic-dominated areas for the pathogen, for Phytophthora agathodicida. And then the very last thing that I'll show you is some work that I just did with the help of Alana Thurston, looking at the degree to which phylogeny or plant provenance can predict the inoculum potential of different species. So what you're looking at here is a phylogeny of 108 species that I tested. You don't need to look at the species names, but the red ones are exotics, and the black ones are the natives. And these bars here indicate pathogen growth on leaves. I did a detached leaf assay, so put a little bit of inoculum on each of the leaves. I measured the growth of the pathogen on those leaves, and we found that exotic plants, the pathogen grew, Phytophthora grew faster, and the lesions were larger on these exotic leaves. So this may be good news for the natives, but it's pretty bad news considering these exotic plants may be strong reservoirs for this pathogen, which is terrible news for kauri. And so just to summarize both of these projects, invasive plants may be important reservoirs for pests in Aotearoa New Zealand. And the ability of exotic plants to tolerate pests and share them with natives may increase their competitive ability and their threat to conservation. And so with that, again, even more thanks to more people. And thank you so much for listening.